Well, aloha everyone, and welcome to my lecture on the reproductive systems. Today we'll be talking about the male and the female reproductive systems, the purpose of which is the end result of either sperm or egg, which are known as gametes. These guys are going to be haploid. That means they're going to have half of the genetic material of both mom and dad, which when they come together to create a zygote, that's during a process called fertilization, they are going to then fuse and restore that genetic complement so they become diploid again. This is going to result in the creation of genetically unique offspring, so they're going to be genetically unique from both their siblings and either parent. And while it's actually very interesting because the male and female reproductive organs are actually very distinct if you look at them anatomically. However, they arise from the same cell population during the process of embryogenesis. And we will be lightly glossing over a little bit of the embryogenesis of the male and female reproductive organs much later on in this lecture. So let's start with the male reproductive system. Some of the organs you may hear me talk about today are going to include the testes, where sperm are actually created and stored, the ductus deferens, which you may know as the vas deferens, the epididymis, which you may not have heard of until right this moment, the ejaculatory ducts, the urethra, seminal vesicles, prostate, bulbourethral glands, scrotum, and penis. So this is an overview of the male reproductive system. It's going to include the reproductive structures and then some other structures you may recognize, for example, the bladder here, which is connected by the peritoneum to the abdominal cavity. Now you'll notice that the bladder actually does share that same urethral flow as the ejaculate, so out of the external urethral orifice through that same canal. So let's start with some morphology that you might understand already. So this here is the penis itself and the testes. The testes are, of course, encased in the scrotal sac here. So this is the scrotum containing the testes and the epididymis. Now, epididymis might not be something that you've heard of before, but it's going to just sit nestled right there on top of the testes, connected both at the caput and at the caudal region. So it has a caput, a corpus, and a cauda, and this is going to be the site of sperm maturation. So sperm are actually created here in the testes, then they're going to come out into the early part of the epididymis completely immature, unable to even swim. We're going to undergo a maturation period of about 14 days where they go through this little tunnel, and the epididymis is basically just a long coil back and forth where they travel very slowly all the way through here until they're ready for ejaculation. When they're ready for ejaculation, they're going to be sent out through the vas deferens, where they're also going to meet up with fluid that comes from the seminal vesicle here. So those two are going to come together and become semen, so that the seminal fluid comes from a different region than does the sperm itself. And when someone has a vasectomy, for example, they're just going to clip this. So they will still have an ejaculate. The ejaculate will only consist of the seminal fluid from the seminal vesicle, and it will not have sperm in it. Now, in a normal situation where we do have a intact vas deferens, both the sperm and the seminal fluid are going to mix together at the time of ejaculation, and they're going to be excreted out through this urethra. So the urethra actually has three separate sections. The prostatic urethra, which is here and is actually going to go through the prostate. 1, 2, 3, right? That's all the prostate. And then as we head through the prostatic urethra, we're going to enter into here the intermediate urethra. The intermediate urethra is going to pass right on through to the actual penis itself, and then this here is going to be called the spongy urethra because it goes through the spongy corpus spongiosum penis and then out through the external urethral orifice. So that is going to be the route of the ejaculation event. Some other organs that I didn't mention here are going to be, let's see, okay, I mentioned those. So the actual body of the penis, so this whole thing is the body of the penis, which is comprised of multiple different sections. This section is the glans penis here, and inside we have the corpus cavernosum penis and the corpus spongiosum penis. And we actually have two of those and one of these, so at a cross section it's going to appear to be two circles of the corpus spongiosum penis on the bottom section closer to the testes, and one of the corpus cavernosum penis here on the top. So this is obviously an erectile organ, so that's all going to come from blood capillary and circulation into this organ, but we will get to that in just a little bit further. Okay, so this is what it looks like in an actual animal and not just a depiction. So these are the seminal vesicles and the vas deferens. They've been severed. That's all going to head out into the prostatic urethra, through the ejaculatory ducts, headed out to the intermediate urethra, and then out here through the spongy urethra to the exit out through the glans penis. Okay, So what is the point of the reproductive system? We have several different sections. One of the main parts that we will talk about right now is the scrotum. So the scrotum is actually a sac of tissue. It's loose skin and sub-Q tissue, and it's going to contain the testes inside. And this is important because the testes actually need to maintain a different temperature than the rest of the male body. They need to be a little bit cooler in order for normal sperm differentiation and creation. Inside the testes, inside the scrotum, it's going to be separated into two separate compartments, and it's separated by something called a dartos muscle and that subcutaneous layer. So this here is the dartos muscle, and that's responsible for contraction of the testes up into the inner body cavity if the men get too cold, or descent if they are too warm, so that the sperm can end up maturing at the correct temperatures. Let's see. So this is what I was talking about when I said the corpus cavernosa penis is here. We have two of those, and then here the corpus spongiosum penis, we're going to have one of those. This here is the ductus deferens, or the vas deferens. It's going to be connecting of the testes to the seminal vesicles, and then out through the ejaculation route. Let's see, what else are we missing here? So this is the cremaster muscle here. This cremaster muscle is going to be surrounding this whole region, including the ductus and the ductus deferens and the autonomic nerves. There's also a testicular artery, some lymphatic vessels in here. So lots of different things inside this region is going to contract up when this individual might get cold. All right, so the testes are oval glands. It's a pair of them. You'll notice, actually, let me go back a second. You'll notice that they never sit directly on top of one another in terms of the orientation, and that's because obviously there's a issue that they might get crushed together if there was some sort of injury event. So typically one testes will be a little bit higher or a little bit lower than the others so that they can orient themselves together in the event of having get caught in a small space. So the testes are going to be found in the scrotum, covered by something called the tunica vaginalis. Inside that tunica vaginalis, there's a region that's made of connective tissue. It's a little capsule. It's called the tunica albuginea. And that's going to extend inward, and it forms like septa, or a division that's going to compartmentalize. And inside the testes, we have something called seminiferous tubules. And seminiferous tubules, really, it's just a very long tubule. They're going to create the sperm and carry them through until they're going to enter into the epididymis. And the creation of sperm is a process called spermatogenesis. And this is what a sagittal section of a testes would look like. So again, this is called the spermatic cord. This is where the cremaster muscle is going to be in control of. Inside that spermatic cord, we have a lot of important things like the blood vessels and the nerves, also the vas deferens, right? So that vas deferens is going to be the route by which sperm are going to end up heading out into the epididymis, from the epididymis, out of the epididymis for ejaculation. All right, so there's a lot of things going on here. So let's start here. This is going to be the testes, and then sitting on top of that here is the epididymis. And you'll notice that it looks like it's all one long tubule, and that's because, I mean, it is. There's a couple of them, but realistically, it's just a very long tubule whereby the sperm are going to travel from one section to the next and enter into different stages of maturation. It's separated out into different lobules, right? The testes here are separated into different lobes or lobules, divided by septums, so different dividing sections. Again, on the outside, this is the tunica vaginalis. Underneath that, we have the tunica albuginea. And then once the sperm have been created in the testes, they're going to head into something called the rete testes, and then from there into the epididymis. They're going to head into the head of the epididymis. They're going to spend about 2 weeks traveling through epididymis. During epididymal transit, they're going to gain a maturational function, so they're going to gain the ability to swim, for example. So previous up in here, their tails are not going to be moving. Down in here, they're going to be capable of fertilizing the egg. These guys are capable of fertilizing the egg only through something like ICSI, or intracytoplasmic sperm injection. They wouldn't even be viable for something like in vitro fertilization, because even in vitro fertilization, the sperm still have to swim towards the egg itself. So if you were going to do something like IVF, you would want to take them from here. I'm off topic. All right, so coming out of the caudal epididymis, you're going to head up into the vas deferens and then up through the spermatic cord. And this is what this looks like in an actual organism and not just in a diagram. You can't really see much of the seminiferous tubules here. This is the testes. The testes, again, is held in place by the tunica albuginea, and around that, the tunica vaginalis. and then this here is the spermatic cord itself. If you aren't actually looking at a cross-section, you've just pulled back this layer of the scrotum itself to take a look. This is the testes inside. The testes, again, has a tunica vaginalis, and then on the outside of it, so we haven't cut into it at all. And then this is the epididymis. Again, the epididymis, this is the head of the epididymis, and then it's going to head down. So this is the caput, the corpus, and the cauda, or the tail of the epididymis, and then this is where sperm will have gained maturation. They've taken about 2 weeks again to transit through here, and then they'll exit out through the spermatic cord during the process of ejaculation. Again, they'll be heading out through the vas deferens. But let's back up a second. Let's talk about spermatogenesis. So spermatogenesis is when we are creating sperm. And we're going to start here with something called spermatogonia. Now, spermatogonia are still diploid. Please remember that we're trying to get to the point where we're haploid. So these guys are stem cells, and they're going to be able to differentiate basically into forever, right? They'll differentiate multiple different times perpetually. And what they create are these primary spermatocytes. These guys are also still diploid, all right? So we have to undergo one more division before we can get to the point where they're haploid. Okay, so this is going to be, if we're looking at a seminiferous tubule, this here is the interstitial cells on the outside. Here's the blood capillary, right? And then if you're going across this basement membrane, now we're entering into the actual testes itself. Seminiferous tubule is going to be comprised of these guys here, the spermatogonium. These are the ones that I was telling you are this is single spermatogonium, plural spermatogonia. They're going to be diploid. Let me take a second to back up because I want you to recognize what this 2N and this 1N is representative of. So when I say diploid, I mean that they have 2 copies of every chromosome in the body. And for humans, that means that we have 23 chromosome pairs. That means we would have 46. But that differs for other organisms. For like mice, for example, it's 19, and so we don't want to go back and forth with the number of chromosomes total. We just want to say it's a full set, which would be 2 of each, or it's half of a full set, which would be 1 of each. And that's what that 2N and 1N is representing. So when I say diploid, I mean 2N, meaning we have 2 copies of each. And that's what happens here in the stem cell and here in the primary spermatocytes. But we have to have another division that gets us down to N, and is going to be haploid. And haploid is what we're going to need because the oocyte also is going to have to be haploid in order for us to come together later on during fertilization and make a genetically unique diploid individual again. Okay, so now that I've made that clear, every time that you see 2N, I want you to think diploid. Every time that you see 1N, I want you to think haploid, or half. All right, so what is inside this tubule, the seminiferous tubule? Here we have the spermatogonium. Again, this is going to be our stem cell. It's going to enter into a primary spermatocyte, right? It's actually going to differentiate into primary spermatocyte. At the same time, it's also going to create another copy of itself so that we're going to be able to sustain that cell line forever. And that's kind of a hallmark of stem cells, is that they always create one that replaces them and then one that goes on to differentiate before they enter into differentiation. But I digress a little bit. What else do we see here? So this here is the blood-testes barrier. It's a tight junction. And when you think of the blood-testes barrier, there's very little fluid that's going to be able to get across here. And this is very important. It's similar to like the blood-brain barrier. But what I want you to know is that it's actually being held together here by what are called Sertoli cells. And they're not actually shown here. They're shown here in these like kind of orange, dark orange color. But these Sertoli cells are kind of nurse cells. They're responsible for making sure that these spermatocytes have everything that they need as they're going through the different stages of differentiation. And so one Sertoli cell next to the next Sertoli cell makes these tight junctions that become the blood-testes barrier. Okay, well, I think I have hammered that diagram home, so let's move on. So the first thing that has to happen, right, we have this guy here, the spermatogonium, that has to create this primary spermatocyte, right? We are still diploid, though, so that primary spermatocyte has to undergo meiosis 1. Now, if you're not familiar with meiosis, the point of meiosis is to make haploid, genetically unique cells, and that's going to be these secondary spermatocytes. So the reason that they're called secondary spermatocytes is because they've undergone that secondary division, which makes them haploid. And then we have a secondary set of division, meiosis 2, and those secondary spermatocytes are actually going to become 4 spermatids. And this kind of confuses people because these 4 spermatids are also going to be haploid, but that's really because in genetics, you know, if you look at a chromosome and you see an X, that's actually two chromosomes one chromosome about to become two. And that X is considered one chromosome, and that X splitting into two I's, now it's considered two chromosomes. So we have all the genetic material there, meiosis 1, so that secondary division is still going to create haploid spermatids. But I digress. My point is that after meiosis 2, you end up with 4 spermatids that are haploid and genetically unique. But here's what's kind of cool about them. So let's look here, these spermatids. These spermatids are sharing information across what's called a cytoplasmic bridge. And from a genetic perspective, this is interesting because this means that they're going to become genetically equivalent, even though this one may have some sort of enhanced ability or less of something than the others, because they're all sharing all information. So a deficiency of something isn't going to affect any of the individuals because the others are going to compensate for it. So it kind of gives them kind of like an even playing field, even though some may have more of an advantage than another. And that's going to end actually as soon as we end up into the spermatozoa stage. But let me run this whole diagram over starting at the top. So here we have the spermatogonium. It's 2N. And remember, every time that we are going to enter into the process of differentiation, which is what's happening here as we enter into this set here to enter into the primary spermatocytes, we're also going to have a reserve set, a precursor set that's going to regenerate that population of stem cells. So this stem cell is going to divide. It's going to have one set that stays exactly the same as itself, and then one cell that's going to enter into differentiation. That differentiation is going to lead to it becoming a primary spermatocyte. This primary spermatocyte, again, is still diploid. It's still 2N. There's a couple things that it wants me to talk about here. One is that we have made sure that the DNA is replicated. We have tetrads that are formed, and something called crossing over, and this is what I'm going to kind of hammer into. Crossing over has occurred, and what this means is that the genetics from the paternal lineage and the maternal lineage have shuffled a couple of times before they're actually going to separate out into the next individual spermatids. And that's kind of cool because that means that has happened at every single time that sperm and egg were created, which means that every single chromosome that you've inherited is shuffled not just from your parents, but also your grandparents and your great-grandparents and your great-great-great-parents. And so in that way, you are actually able to inherit all of these different features from multiple different people in your lineage. But again, I digress. All right, so here we have the crossing over event. Again, we have shuffling of genes. And then we have another event that occurs that's meiosis 1. And this is when we're going to go from 2N to N. So we're going to go from diploid to haploid, right? And then that haploid secondary spermatocyte is undergoing the second set of meiosis 2. It's still maintaining haploid even though it's dividing from 2 to 4, which I know is confusing, but just believe me. And then we have these spermatids that are sharing information via these cytoplasmic bridges and creating eventually these spermatozoa. Another thing I want to point out here is that these sperm have very, very tight, small little heads. So if you look at normal cells, you see a nucleus, and you see cytoplasm around it, okay? The cytoplasm is where all of the important activities take part. That's not true. The nucleus is where all the DNA is housed, and that really is the most important part. But the actual action is occurring in the cytoplasm. That's where proteins are being made, etc. And so if you look at these sperm cells, not only do they have no cytoplasm at all, but they also have very compact, very tightly compacted DNA in these tight chromosomes that are in these really small nuclei. And so what that means is that the DNA inside these sperm is not being processed anymore. These guys are not making RNA, they're not making protein, they're not doing anything themselves. So from this point forward, because they don't have the ability to have the normal DNA makes RNA makes protein functions, they're going to have to be taken care of by all of the cells around them. And that's where the Sertoli cells or the nurse cells around them come in. We'll talk about that in just a short while. All right, so let's talk about the different parts of the sperm. So the objective of the sperm is that it's going to be able to fertilize an egg, right? It has to get through this cumulus mass, so it has to penetrate through this region of the oocyte that is going to be kind of a barrier, and there's going to be a whole bunch of them doing this at the same time. It's a cumulative effort. One sperm is never going to be able to do it by itself. But it's a race to achieve this fertilization event, and the one that actually hits that zona pellucida, or that barrier first, is the one that's going to be responsible for creating the zygote with the egg. And the rest of them are all going to be, thank you very much for your time, have a nice day, sent home. We have several regions of the sperm. The sperm head is going to be what I just mentioned, has the nucleus, so really tightly compacted chromosomes inside that nucleus, and then there's very little cytoplasm around there. And, well, there is very early on, and that's called a cytoplasmic droplet, and it's shed off halfway during sperm creation. And it's actually a marker of sperm that are maybe immature or haven't quite gotten to the point where they're able to fertilize the egg because they still retain their cytoplasmic droplet. But I digress. A normal sperm that's ready to fertilize has very little cytoplasm left. On top of the head, we have something called the acrosome. And the acrosome kind of looks like a tiny little balloon that has pushed over top of the head and has these specialized enzymes. And these enzymes are going to help with penetration through the cumulus mass, which is one of the very last barriers that the sperm have to penetrate prior to zona pellucida, which is the actual, zona pellucida binding is the actual fertilization event. From that point forward, there's an event that occurs that makes it so that not any more sperm are able to fertilize from that point forward. So we make sure that we only have one sperm entering into the egg at any point and never two. So past the head, we have the neck. The neck has centrioles, and the centrioles are going to be connected to the microtubules, and the microtubules are going to make up the rest of the tail. And if you know anything about sperm, you know that sperm swim, right? And that's because of these microtubules inside the tail that are sliding back and forth across each other that are making this S-shaped motion, because one pulls as the other pushes, and vice versa. Well, I guess they just pull at opposing times, but that pull pushes the other at that moment. Anyway, inside that tail, we have a middle piece and also a principal piece and an end piece. So the middle piece is going to have the mitochondria that makes ATP responsible for locomotion of the sperm. So sorry guys, during the fertilization event, all of your mitochondria are lost. This is why all of your mitochondrial DNA follows your maternal lineage, because it only ever came from your egg and from your mother's egg and from her mother's egg all the way on, because the fertilization event, only the head gets in and none of the tail does. So inside the principal piece and the end piece, we are going to also have these microtubules, and those are going to be a little bit larger at the principal piece and a little bit smaller at the end piece, but that's going to be responsible for the tail motion. And that looks kind of like this. So as I mentioned, we have two major regions, the head and the tail. Let's talk about the head first. So the head has this region here that is the nucleus, and as you notice, there's very, very little cytoplasm, right? It is the nucleus that is sheathed in an acrosome, and I kind of call this like a balloon. So if you picture like a kind of deflated balloon that you push your hand into, the acrosome is going to sit over top of that. And inside here, we have these hyaluronidases, and hyaluronidases digest hyaluronic acid, and hyaluronic acid is what connects the cumulus cells together. So the cumulus mass that the sperm have to penetrate through prior to binding to the zona pellucida for fertilization to occur is going to have to be digested by the enzymes that are inside this acrosome. So as you can see, one acrosome isn't going to be enough. It's going to be a cumulative effort of multiple different sperm having what's called an acrosome reaction, or a release of these hyaluronidases at the specific location, so that we can then tunnel through the cumulus mass, and eventually one lucky sperm will end up binding to the zona pellucida. And most likely the strongest sperm, which is obviously the point from an evolutionary perspective. All right, so we're moving our way down here. This section is the tail. The head is connected to the tail by the neck region. The neck is very small. And then the tail section is going to be where all of our mitochondria are here in this, this upper region here, this middle piece. Then we have the principal piece and the end piece. And running through here, you can see this line here, those are the microtubules. Those are going to shimmy back and forth, and then that's going to make the tail go back and forth and make a nice little S motion that allows it to travel directionally, as opposed to something like cilia, which allow fluid to travel over top of it. This would allow the sperm to travel through the fluid. But I digress again. Okay, so let's talk a little bit about hormonal regulation. So once men hit puberty, we have something that's called gonadotrophin-releasing hormone, or GnRH. And that's going to stimulate the anterior pituitary gland to make luteinizing hormone and follicle-stimulating hormone. So you've seen this, you will see this later on in the female reproductive cycle as well. So people often think that it is responsible only for females, for things that happen in the female reproductive system, but that's not true. It's actually going to help the cells in the testes produce testosterone, and follicle-stimulating hormone also is going to stimulate spermatogenesis. So although follicle-stimulating hormone was named that because it stimulates the follicles in the female system, it's also involved in the creation of sperm, which is not necessarily all of that surprising considering that these cells did come from, or these organs did come from the same cell population much earlier in embryogenesis. So it doesn't surprise people that they are under the similar sets of hormonal regulation. Okay, there's a lot going on here on this slide. So this here is the hypothalamus and the pituitary. So no, this is not the testes, guys. All right, so here's the hypothalamus. The hypothalamus is going to be responsible for the creation and secretion of proteins into the pituitary gland, the anterior and the posterior pituitary gland, little region there called the infundibulum, and that is going to be responsible for creating FSH and LH. And as I just mentioned, FSH, it's also going to need the help from testosterone, is going to create sperm, so it's going to induce spermatogenesis. Okay? So here's testosterone heading in with FSH. LH, again, with testosterone. So LH is going to stimulate the secretion of testosterone, and the testosterone is going to do a couple of things. So the testosterone is going to, as I mentioned, accumulate here with FSH to stimulate spermatogenesis. It's also going to go back and forth into dihydrotestosterone. And the testosterone is going to decrease the release of GnRH and LH. So it's actually going to be a negative regulator of its own creation, if that kind of makes sense, at both the hypothalamus level and also at the anterior pituitary level. So you may recall the hypothalamus creates regulatory molecules, hormones, etc., and then they are stored in the pituitary where they are later released. So there's a couple steps of regulation here, and so it targets both of them. The testosterone targets both the hypothalamus, which is creating GnRH, and also the anterior pituitary, which is releasing it. So that's going to decrease the entirety of the GnRH in solution. All right, so let's look over here. These are spermatogenic cells. They're going to be creating sperm, and then again, that's going to happen because of follicle-stimulating hormone and testosterone coming together. But one of the things that they're going to also create is inhibin. And inhibin is going to decrease the release of follicle-stimulating hormone. So it's also going to be its own negative regulator. So we have a couple sets of negative feedback loops in this tightly regulated hormonal regulation. All right, so here, talking about dihydrotestosterone, just a little sidebar, it's going to induce the male pattern of development, so this is going to be important before birth. It's also going to be responsible for getting those secondary male sex characteristics, so the changes that occur at puberty. And it's also responsible for protein synthesis, so anabolism. You may remember that metabolism is comprised of catabolism and anabolism. So anabolic steroids pump you up like Arnold Schwarzenegger. All right, so anabolic pathways build things, and catabolic pathways break things down. So protein synthesis is an anabolic pathway, and dihydrotestosterone, or DHT, is responsible for this. So this is one of the things that can go wrong if we do have like gender dissociation, where a person is presenting with a different phenotype than their genetics, right? They're presenting as female even though they have a Y chromosome, for example. Because they may have issues with this particular pathway, and that's going to be very important with the organ development before birth. There's a lot of other things that can go wrong, and we'll touch on a couple of those possibly as we head through here, but anyway, this is kind of an overview of some of the hormonal regulation of the male reproductive system. So testosterone and dihydrotestosterone, so that's what we were just talking about here, this DHT, right? They kind of go back and forth, and they are going to be responsible for multiple different things in male development, including the prenatal development of male sexual organs, and also the development of male sexual characteristics, and then later on also development of male sexual function, and again the stimulation of anabolism. And so this is one of the reasons why men tend to put on protein mass much more easily with exercise, because they actually are going to have higher levels of testosterone, so they're going to be able to put on muscle mass. It's also associated with anger and aggression, as a side note, or it can be. And there's a negative feedback system that controls the blood level of testosterone. Again, that's not surprising because we've seen a lot of these negative feedback loops before, but just to kind of refresh you, again, we're going to have a homeostatic condition that is disrupted in some way or another by increasing or decreasing whatever that controlled condition is by a stimulus. So in this case, we have a stimulus coming in that is disrupting homeostasis by increasing, in this case, the blood level of testosterone. So I have a normal homeostatic condition of blood testosterone, and all of a sudden we end up with an increase of our testosterone levels. So just like anything else in our body, we have certain receptors that detect the levels, right? These chemoreceptors that detect the levels of certain chemical compounds, including testosterone. And so we have receptors in the hypothalamus that are going to create that GnRH that we talked about previously. And so when we secrete more of the GnRH, that is going to end up activating information in our control center, which is where our anterior pituitary gonadotrophs are. So remember, we have to create the hormones, and then we also have to secrete them. So they're created here, they go through the infundibulum, and then they're stored here in the anterior pituitary. And so here in the anterior pituitary, we have these gonadotrophs that are going to be responsible for the secretion of these different cells. Anyway, we're going to end up with a decrease in the luteinizing hormone, or LH, in the systemic blood, which is going to affect the Leydig cells of our testes. And I haven't necessarily talked about the Leydig cells. The Leydig cells are interstitial cells that are found in between the Sertoli cells and the spermatocytes and the developing sperm. And these Leydig cells are responsible for secreting testosterone, so they're part of the testosterone response. So if we are decreasing the levels of testosterone that's coming out of these interstitial or Leydig cells, that's going to decrease the blood levels of testosterone, which is going to restore homeostatic conditions. Again, in this case, we had higher levels of testosterone than needed, so by turning testosterone off, we have resumed homeostasis. Okay, so previously I mentioned that we have all these tubules and ducts, and the male reproductive system is actually just a series of tubes that are ways of transporting fluid and cells within that fluid, such as sperm, from one part to another. So sperm and fluid are going to travel from the seminiferous tubules of the to the straight tubules, and then into the rete testes. And from the rete testes, then they're going to go into the epididymis. And as I mentioned, the sperm are going to spend about 2 weeks in the epididymis. They're going to mature there. They're going to gain fertilizing capacity. They're going to have an increase in their acrosomal enzymes. They're going to lose their cytoplasmic droplets. A lot of things happen here. They're going to gain motility too, so they're going to be able to swim from this point on. So oftentimes when they're going to be doing in vitro fertilization, they want to make sure that they're taking the mature sperm from the very lower sections, the caudal regions of the epididymis. But if sperm don't actually end up having the ejaculatory event, taking them out through the, then they're going to end up degenerated and reabsorbed inside the caudal epididymis. But on the chance that they are a lucky set of sperm, they're going to be propelled out through the epididymis into the vas deferens. From the vas deferens, they're then going to meet up with fluid from the prostate, from the seminal vesicles, etc., and then they're going to end up out through the ejaculate. Okay, so let me just reorient you. I know you've seen this figure before, but I'm going to, now that I've talked to you about what happens, maybe it'll kind of help you a little bit. So again, we have these different lobules with the septums that divide them, and inside them we have these seminiferous tubules. They're very long tubules, and this is where the sperm are created. So spermatogonia are going to end up in primary and then secondary spermatocytes, and then end up being sperm. They're going to be immature sperm at this point, but they're all going to come from these different lobules, right, into this rete testes, and from the rete testes into the epididymis. Now from the epididymis, this is one super long coil that they're just going to end up traveling, and as they go through it, they're going to end up undergoing different stages of sperm maturation and development, kind of like the way you would go through as a cohort in your university. They do all kind of the same thing, so when they come out on the other side, they've all matured together. So they're going to head into the head of the epididymis. Again, this is the caput, this is the corpus, this is the cauda, and then here they're going to wait for ejaculation. If ejaculation does not occur, or say, for example, ejaculation occurs but they are not needed, for example, in a vasectomy where we have eliminated this vas deferens, So we're still going to have ejaculation from the prostate and the seminal vesicles. It simply will not contain sperm cells. Then the sperm are going to be resorbed here in the caudal epididymis. Okay, so just to kind of refresh you with what we've just talked about. So the vas deferens, or the ductus deferens, is going to head out through the caudal epididymis, or the tail of the epididymis, and head up through the spermatic cord that heads up into the pelvis. It's going to go over top of the ureter, right, which is part of the urinary system, passes over the side and down the posterior surface of the urinary bladder. So it truly does have quite a long way to go before it meets up with the rest of the fluid that's going to be ejaculated. And that's going to include things that are sent out from the prostate and from the seminal vesicles. Okay, so the vas deferens, remember, was made all the way down here. It's going to go up, it's going to go over, and then it's going to come back down, right? The vas deferens, and then past here to be able to meet up with fluid from the seminal vesicles, etc. So it has quite a long way to go, and all of this is going to be situated all around the urinary bladder, right? And here we have a pretty good view here. This is the prostatic urethra here. This here is the prostate. So you can see why if you had an issue with an enlarged prostate or early stage prostate cancer, you might have issues where you had a full urinary bladder and the urge to urinate, but unable to relieve yourself because you had pressure directly on that urethra. This can also cause issues with ejaculation as well, where the fluid is attempting to go forward and ends up pushing backwards. So it can be quite painful. But I digress. So this here, again, this is the ejaculatory duct. Those are going to end up ending up secreting into the urethra, and then from the urethra, again, we have three separate sections of the urethra: the prostatic urethra, the intermediate urethra, and then the spongy. So here prostatic, here intermediate, which is very short region compared to the other two, and then the spongy urethra, which is obviously the longest one because it's going to extend to the length of the penis itself. Okay, so that's kind of an overview of the male reproductive system in terms of the transit of the sperm, etc. So let's take a step back and let's talk about the anatomy. So we have that spermatic cord, again, that's going to come up from the scrotum. It's going to contain in it the vas deferens, but it's also going to have a lot of vasculature. So it's got the testicular artery, veins that are going to be coming out of the testes, also a lot of nervous system, right? So autonomic nerves and lymphatic tissue as well. And it also has that cremaster muscle, right? The cremaster muscle is going to be responsible for pulling the testes up if you end up cold or injured or something like that. So that spermatic cord is also going to run right alongside the ilioinguinal nerve. And that's going to pass through the inguinal canal, which originates at the deep inguinal ring and is going to end up ending at a region called the superficial inguinal ring. And let me show you what I'm talking about here. Okay. So here we have, so this is the testes. We talked about previously. We have the tunica albuginea of the testes, and then the vaginalis. Here we have the dartos muscle, which is going to extend right around and cup the bottom of the testes. Here we're going to have the spermatic cord. Inside that spermatic cord, and it's going to lie right along this cremaster muscle, which is responsible for pulling it up if we want to have the testes ascend for a reason. Alongside there, we're also going to have the ductus deferens, or the vas deferens. Again, this is going to be a route of sperm that are going to come out of the testes and then head all the way back out and around to be able to come out through the ejaculatory canals or the urethra. We also have testicular arteries and testicular veins. We have autonomic nerves. So a lot of stuff is hanging out inside of this spermatic cord, right? It's quite important. Now, if we look on the other side and we're looking at the actual, the muscle itself, we have a lot of muscles and a lot of ligaments that are going to be connecting the penis to the rest of the body. So here we have the internal oblique muscle. This is the external oblique muscle. It's been cut off so we can be able to see what's going on. We have some ligaments that are going to hold the top of the penis in place. That's going to include the fundiform ligaments here, right? It's going to kind of surround the suspensory ligament of the penis. And then if we are going to be looking at a transverse section of the penis, so as I mentioned, we have two sets of the corpus cavernosum penis. Those are those top two rings here. And then the spongy urethra, which is going to be surrounded by, so the urethra is the canal by which we're going to have urine or ejaculation exit. It's going to be surrounded by the corpus spongiosum penis. All right. So The ejaculatory ducts are going to be from two different sets. They're going to come from the seminal vesicles, right? So it's going to be a duct from the seminal vesicle and also a duct from the vas deferens or the ductus deferens. So the seminal vesicle is going to have things that are going to help keep the sperm happy and healthy during the transit through the vaginal canal and then through the cervical mucus and all of these different barriers to fertilization that they're going to have to transit. And the vas deferens is going to carry the actual sperm cells themselves. So semen is going to be comprised of the seminal fluid and also of sperm. Okay, so the urethra is that duct, that tube that's going to be shared by both of them, both the reproductive system and the urinary systems. So both semen and urine are going to be able to pass through it, obviously not simultaneously. It's going to pass through the prostate gland, so we have several different sections, right? Remember, we have the prostatic urethra. Then it's going to pass directly through the muscles of the peritoneum, where it's going to go from kind of the inside of the body to, it is still inside the body, but now it's inside the penis on the outside of the body, and that happens at the exchange of the intermediate urethra. And then the later part is going to head through the penis itself, and that's going to be called the spongy urethra. And I know I've hammered that home a couple of different times, but now you can see it for yourself. So here's what this looks like if we're going to be taking a frontal section. So here's the urinary bladder. The bladder is going to head right into the urethra, right, just for regular urination, but also heading into that urethra, we're going to have the ejaculatory ducts. The ejaculatory ducts are going to come from, again, from the seminal vesicles and also from the vas deferens. This here surrounding that, here is the prostate. And we're going to head through the peritoneum, and that's going to be when we change from the prostatic urethra through the intermediate urethra. Again, that's only for a heartbeat until we enter here into the spongy urethra, which is going to travel out through the external urethral orifice, so through the entire body of the penis. And again, the body of the penis is going to be comprised of corpora cavernosa penis and corpus spongiosum penis. And then we also, here it's showing a foreskin as well. So we have the body of the penis, and then here at the bottom we have the corona, which is going to be the bulbous part at the lower section of the glans penis. At the top part of the glans penis, we're going to have the external urethra orifice. All right, so when we think about the reproductive system, we really typically just think about sperm, but there's a lot of other things that have to go into that seminal fluid. So some of the accessory glands include the seminal vesicles or the seminal glands. They're going to secrete a very viscous fluid. It's a white viscous fluid that's going to contain a lot of sugars. Fructose, also a lot of things like prostaglandins and different sets of clotting proteins. So there's actually a lot of different things that the seminal vesicles are going to secrete that are going to be kind of a last-minute assist to sperm fertilizing capacity. You can picture this like sending these guys out with a packed lunch for a long day at work. The prostate is going to be, it's a donut-shaped gland, and again the urethra is going to run right on through it, and it's going to secrete a milky fluid. It's slightly acidic, so you'll note that we have one that's alkaline and one that's slightly acidic, and that's going to contain citric acid and then a ton of proteolytic enzymes, so things that are going to break down proteins, because one of the first things that's going to happen to these sperm is they're going to be kind of under attack in the female reproductive system. So these are going to be things that are going to help them in their journey. We also have something called seminal plasmin and acid phosphatase. All right, so Cooper's glands. Cooper's glands are also known as bulbourethral glands. These are going to make an alkaline fluid, and it's a fluid that is secreted early in sexual arousal that is going to help neutralize what's already in the urethra and also partially help protect against certain STDs. So it's going to, particularly bacterially transmitted ones, because it's secreting an acid. It's an alkaline that is combating the acid. And so it's going to help neutralize the acid from the urine and also mucus, and it's also going to create lubrication. All right. So semen, as I mentioned, is going to be a mixture of sperm, which are the cells themselves, and then seminal fluid, which is going to be comprised of what comes from the seminal vesicles and then also from the prostate. And the volume that an average ejaculate is approximately going to be 2.5 to 3 milliliters. And it has approximately 50 to 150 million sperm per milliliter. Normal pH is going to be 7.2 to 7.7, which is normal pH of the body, but do note that the vagina tends to be mildly acidic. So one of the first things that it's going to hit is going to be vaginal acidity. So the sperm are going to end up having to combat that, which is why the seminal fluid is going to be lightly alkaline. just a little bit higher than the blood, right, which would be about 7.5. All right, so inside the penis, we have the urethra, and the urethra, again, is that tube. It's a passageway for semen and urine, and it has 3 separate cylindrical masses, 2 of which are the cavernosa penis, so the corpora cavernosa penis, and then one that is the corpus spongiosum. And all of these are erectile tissues. They're all going to be tissues that are going to be vasculature, highly vascularized, and also a lot of nervous tissue, so highly sensitive tissue. Then at the very tip of this, we're going to have the glans penis, which is the head of the penis, and in an uncircumcised male, this is going to be covered by something called the prepuce, or we may also know it as the foreskin. Okay, so if we are going to take a transverse section of the penis, so here we're going to see, I'm just going to kind of do the overview, and then we'll take a superficial, or we'll do an overview, and then we'll take a more detailed look. Here we have 2 sets of the corpora cavernosa penis, and this is the corpus spongiosum penis. You'll see these arteries. These are highly vascularized tissue. Again, it's going to be responsive to vasculature in order for the stimulation and ejaculation events to occur. We also, around that, are going to have the tunica albuginea that's going to surround the corpora cavernosa. Right, and if we take a look here at the very top here, so at the dorsal section, we're going to see here that we have these deep dorsal veins, and superficial dorsal veins, and dorsal arteries. So we have a lot of vasculature to these regions. In fact, these are so large that you're able to actually see them in cross sections, right? And you can see here that this is going to be kind of an area that can fill with vasculature, right? And if it fills with blood, that's going to cause us to be taut and ridged and create the erection. Okay? So what holds the penis in place? And it's going to be suspended by ligaments, just like almost all of your internal and external organs. It's going to be supported by ligaments. And so we have the fundiform ligament and the suspensory ligaments. As I mentioned, we also are going to have innervation and vasodilation occurring, and they kind of have to occur in concert. So a lot of things have to happen in order to get a stimulated erection. So it's going to be caused by parasympathetic innervation, so it's not going to be helpful with the fight or flight response, right? And it's going to end up with the vasodilation of arterioles and erectile tissue. So in order for the blood to be directed there, it's going to have to be directed from other places, which is why it's involved in the parasympathetic responses. So large amounts of bloods are going to pool into this tissue from these sinuses that are going to be widely dilated. Right? And then the process of ejaculation is how we're actually going to be releasing semen. Remember, semen is going to be comprised of sperm and seminal fluid, so not just the sperm cells themselves, but also all of the fluid that accompanies them to send them on their journey. And this is going to be in response to sympathetic stimulation. And so this is going to be a very strong contraction, and it's going to be the responsibility of multiple muscles to be able to force the semen out. That's going to include the bulbospongiosus, the ischiocavernosus, the superficial transverse peritoneal muscles. These guys are going to work in concert to be able to contract, which is going to force the semen out. Okay, so let's take a breather now. We're going to switch topics a little bit. We're going to talk about the female reproductive system. Okay, so it's a little bit more complex because it's not happening perpetually the way the creation of sperm is happening perpetually. It's happening on a cycle that is hormonally regulated, and it also only happens during a set window of years. So while men from the moment that they end up entering into puberty are able to create functioning sperm and have sired children into their 80s, although it's obviously lower and lower prevalence, women have a end time period of fertility, which we know as menopause. Okay, but I digress. Let's jump right in with the organs included in the reproductive system of the female. So organs are going to include the ovaries, the uterus, the uterine tubes, which we may know as fallopian tubes. You may also know them as oviducts. Those all mean the same thing. So if you hear uterine tubes, if you hear fallopian tubes, if you hear oviducts, all of that means the area between the ovaries and the uterus, that is the tube that the egg drops into prior to entering into the uterus. All right? And then we also are going to have the vagina and the other external organs. So this is going to include like the cervix, right, which has cervical os, etc. All right, so this is what the female reproductive system looks like in a sagittal plane. So to orient you, this here is the spinal column, right? This here is the uterus itself. The uterus is going to be, so we're taking a sagittal plane, so we're looking kind of inside here from the middle out. This here is going to be the uterine tube, which is connected to the ovary, not directly, right? These fimbriae kind of wrap around it, but there's a little bit of a gap that the egg is still going to have to go across to get across from the ovary into the uterine tube. The uterine tube is actually going to be the site of fertilization, so this is where the sperm are going to have to swim up to and fertilize the egg, generally around here. And then the fertilized egg is going to tumble down the rest of the uterine tube and then find a nice warm cozy spot inside the uterus to implant itself post-fertilization. Alright, so this here is the rectouterine pouch. It's going to be the area where the rectum and the uterus are going to be connected to the rest of the body. Here is the vesicouterine pouch. That's over here. It's where it's going to be connected right above the urinary bladder. This here being the urinary bladder. Again, the urinary bladder is going to be secreting your urine out through the urethra. However, in the female, we actually are going to have an entirely different system for our urinary system and our reproductive system in terms of tubes and ducts. So there is no connection between the ureter in the female and the female reproductive system. But there is obviously in the male, as we spoke about a minute ago. Alright, so this is the vagina. The vagina is where the penis is actually going to enter into. At the top of the vagina, we're going to have the cervix. The cervix is all the way up here. So one of the first barriers that sperm are going to have to get through is going to be the vaginal mucus. And the vaginal mucus is going to be sitting, well, first of all, it's going to be a little bit acidic, and then there's going to be a serious amount of it here at the cervix, right, the cervical mucus. Once they get through the cervical mucus, and this is going to wipe a lot of them out, right, not all of them are going to make it through. Once they get through the cervical mucus, then they're going to have to go up into the uterus, right? Once they go up into the uterus, then they have to go over into the fallopian tubes or the uterine tubes. Then they have to fertilize an egg, which has to be there at the right time, which is why fertilization really is such a delicate window, particularly on the female side, because it's a cyclical event with the egg dropping. Okay, so from the external side, it's not shown very well. We'll get a different diagram here, but again, here is the urethra, right? Here is the labium majora. This here is shown as the clitoris. This is the mons pubis, and then this is the pubic symphysis, or the pubic bone. And you can see there's a lot of fatty tissue here. That's all depicted in yellow. And then again, behind that, we're going to have the urinary bladder on the other side of the pubic symphysis. Okay, I think I've touched on everything that we need to there, and if we have any more questions, we can always circle back to it. So the ovaries are going to be similar to the testes. They're actually homologous to the testes. So that means that in the absence of the male differentiation clues, the same cells that would normally become testes, if they were given the male differentiation clues, in the lack of those cues, become ovaries. So ovaries are paired sex glands, right? They are homologous to the testes. They produce gametes. These gametes are going to start off immature, and then they're going to mature into what are called ova, or eggs. It's going to be quite different than the process of creating sperm, because during meiosis for sperm, we create 4. During meiosis for eggs, we create 1, and it's a very large one that takes basically all of the cytoplasm from everything else around it, and then we create 3 that are basically throwaways. We'll talk about that when we get to the female meiosis section. But these mature ova are what are actually going to make it into the fallopian tubes for fertilization. So they're secreted by the ovaries. The ovaries also make hormones, a lot of different hormones, including progesterone, estrogens, inhibin, and relaxin. And there are several different sets of ligaments, just like any internal organs are going to be held in place by ligaments. These are going to include the broad ligament, the ovarian ligament, and the suspensory ligament. And those are going to be here. So here's the broad ligament, here's the round ligament, the ovarian ligament, and the suspensory ligament. So anytime you see something that looks kind of like it's just holding things together, it's probably a ligament. It's just kind of holding something in place. Okay, so here we are looking at the uterus. This is the uterus here. The uterus has 2 opposite sides that are going to connect up into the uterine tubes. And then here are the ovaries. So we have 2 ovaries. These are going to ovulate on alternating cycles. So generally this one's going to be, for example, the month of January, and then the month of February, and then, you know, so on and so forth. So these ovaries are going to secrete 1 egg when it is their turn at that month. And that egg is going to end up fertilized here in the uterine tube, and then travel down into the uterus. Now, if we do have an error where both ovaries are making eggs at the same time, and we have a fertilization event, which can occur from the same coital event, or alternate coital events, depending if they're close enough, then we can end up with what's called fraternal twins. right? So twins that come from two different eggs but embed in the uterus at the same time. Now, biologically, we don't necessarily want that because the uterus is meant to house one. It's not impossible to house two, but obviously risks increase with multiple births, and so it is intended initially, that's why we're alternating back and forth, for there to be one in there. So it is considered kind of an error for the fraternal twins. All right, let's see, what else am I missing here? I think I've touched on everything except maybe vasculature. Here's the iliac artery. All right, moving on. So if we're looking at the ovaries under the microscope, so you're talking histologically, we have the top layer is the germinal epithelium. That's going to cover the surface. Underneath that, we have the tunica albuginea. Interesting, didn't we just hear about that when we were talking about the male reproductive system? Again, we're going to see a lot of similarities because these come from similar tissue types. So the tunica albuginea is a dense capsule that is going to be below the germinal epithelium, and the ovarian cortex is going to be found beneath that. And many times you've seen now cortex and medulla, right? Cortex being the upper section and the medulla underneath that. The ovarian cortex is going to be very unique, though, in terms of other sets of cortex that we may have talked about previously, or cortices I may have talked about previously, because it has follicles. So it has these ovarian follicles that are going to be releasing eggs, and they also are going to have stromal cells. So what is not a follicle is going to be surrounded by stromal cells. The ovarian medulla is going to have a bunch of connective tissue in it, including, it's also going to have blood vessels and vasculature and lymphatic tissue, nervous tissue. So the medulla is basically going to be the supply network for the cortex, which is responsible for creating eggs. So let's talk about eggs for a second. So eggs are found inside ovarian follicles. And when we talk about eggs, we're actually talking about oocytes. And oocytes have multiple different stages of development and maturation. So they have follicular cells and granulosa cells inside the ovarian follicles as well. So it's not just going to be the egg inside there. There's a bunch of other things that are taking care of the egg. Again, anytime you have a specialized cell, you probably are going to need someone to take care of it for it because it has such a specialized function that it can't do regular cell processes. All right, so if the follicle is mature, and usually we only get one a month, really we get one every other month because hopefully the alternate ovary will take over for you. Although we have seen in cases where women have the ovaries removed, she still is going to ovulate monthly. It's that for some reason the other one just takes over, the one that's remaining just takes over and secretes one a month for the rest of her mature lifespan. So she doesn't see a loss in her reproductive capacity generally. All right, so we have a mature follicle. It's ready to rupture, and then it's going to expel out that secondary oocyte. So a secondary oocyte is going to be ready for fertilization. So after ovulation, we have the region that was filled with the egg or the oocyte, and now we have an empty house, basically. It's our empty nest. And that's called a corpus luteum, and that's going to produce a lot of different hormones, including progesterone, estrogens, inhibin, and relaxin, all of which you may be familiar with are going to be responsible for different stages of the female reproductive cycle or menstrual cycle. And so the corpus luteum is actually very responsible for maintaining regularity of the female cycle. Okay, so what does that look like? So we've seen the word hilum before. We saw that when we were talking about the kidney. Hilum is just going to be a region where we have an indentation that's going to be supplied with blood. So we have blood vessels here. And just like we saw with the testes, if we're going from the outside, well, first we have the germinal epithelium. But then underneath that we have the corpus albicans here. And if you're taking a look at follicles that are getting ready to be created, right? So here's the primordial follicle. It is going to mature into a primary follicle. That primary follicle is going to go through a couple more stages and mature into a secondary follicle. You'll notice at this point that the oocyte itself is going to be separated by almost everything else, right? It's going to be completely isolated over in the corner while we have this really nice barrier of the oocyte and everything around it. So we're actually going to end up eventually having this mature follicle with a ton of follicular fluid that's going to end up rupturing. And when it ruptures, it's going to be called a corpus hemorrhagium at the point of rupture. And around it, around this egg, which is going to be a secondary oocyte, is the act of ovulation, by the way. So it's going to be secreted from the ovary, and with any luck, going to get picked up by this uterine tube here, right? The fallopian tubes. but there is the possibility that it won't, and it could alternately get fertilized and end up external, and that's called an ectopic pregnancy. But with any luck, it makes it out, and it comes in here, and then is ready for fertilization. So we have the discharge of the oocyte from the ovary, and then we're also around that oocyte we have what's called the corona radiata, which is comprised of cumulus cells. I may have mentioned the acrosome previously when I talked about the sperm and how the acrosome has to go through a bunch, or the sperm have to go through a bunch of cells, and the acrosomes help with that by releasing this enzyme. This is what they have to penetrate through here, is this corona radiata, or the series of cumulus cells. All right, so now the follicle has ruptured, and it is going to be kind of involuted. It's going to be taken back in, and inside there we have a blood clot. Remember, we had a whole bunch of blood that was feeding this previously, and now it has no purpose, so it's just kind of be resorbing. And we have this corpus luteum here, which is folding in upon itself, eventually degenerating, and is going to be left with this corpus albicans. So that's going to be the overview of what happens to a follicle in its lifespan, right? When it's going to be creating, maturing, discharging the oocyte, and then resorbing back into the ovary itself. Okay, so the formation of gametes itself is called oogenesis. And I'm going to go back a stage. These guys here already have the gametes inside them. These primordial follicles already have the gametes inside of them, and it begins way before a female is born, right? It happens during the process of meiosis. By the time an infant female is born, she already has all of the eggs inside of her that she's ever going to use, right? And it's interesting because that means that a woman who is carrying a female offspring is actually also carrying her grandchildren as their, haploid precursors. But anyway, I'm off topic again. So when we have these primordial germ cells that migrate out of the yolk sac during fetal development, they're going to eventually differentiate again into oogonia, which are going to be precursors to the oocytes. The oogonia divide; they become germ cells. Some of them become primary oocytes, and each of the primary oocytes is going to be surrounded by a follicular cells, and that forms that primary follicle. And again, I'm going to go back one. Here's that primary follicle, primordial follicle here, and it's going to enter into the stages of primary follicle and follicular development, etc., when a woman enters into puberty, so 13 to 15, something like that. So it's basically just going to stay there. In a child, it's going to stay here in this primordial follicle stage until we get the differentiation and development cues of puberty. Okay, apologize, I'm going forward again. This here is the primary oocyte itself, and it's inside the primordial follicle. Right, here's these follicular cells that surround it. Around that we have stromal cells, and those are held together by fibers. These guys are collagen fibers, so this is what a primordial follicle is going to look like. And then it gets differentiation clues. And again, it doesn't get these until puberty, but once puberty happens, and for every month on until we reach menopause, we're going to have the secretion of follicle-stimulating hormone and luteinizing hormone. And these guys are going to stimulate the development of the primordial follicles. Only one at a time is going to reach maturity. So a few of them are going to start to grow and develop into those primary follicles, but then they're going to halt there and arrest as primary follicles until they get the clues to continue forward. And again, with assisted reproductive technologies, we can induce those cues and get a ton of eggs all at once if we wanted to do something like in vitro fertilization. So we want to do an egg harvest and then make several of them in the laboratory. We can kind of alternate those cues, but normally we're only going to be creating one every month. Okay, or maturing one every month, I should say. All right, so here we have the primary oocyte itself, and now we've gotten to the point where it's a late primary follicle. It's about to turn into the follicle it's going to enter into maturation. It's surrounded by the zona pellucida, and you may have heard me mention the zona pellucida previously because this is going to be the very last stage of fertilization when the sperm bind to the zona pellucida. Surrounding that, we have these things called granulosa cells. Granulosa cells are going to surround the oocyte, and then they are, in fact, themselves surrounded by a basement membrane, and then also by what's called theca folliculi, these guys here, and that is also going to be held together by collagen fibers. As we move on through the stages of the development of these eggs, the oocyte is going to become a little bit more complex. It's going to be surrounded by multiple different layers of columnar cells. These are called granulosal cells. They're eventually going to become the cumulus mass. And the zona pellucida is going to form between the primary oocyte and the granulosa cells. And it's made of glycoproteins, and it's basically going to be like a large fence that surrounds the whole primary oocyte and is going to be the very last barrier to fertilization. So as soon as the sperm binds to the zona pellucida, the head of that sperm gets to go through, and then a quick reaction occurs that prevents any other sperm from being able to bind. So it goes from being a chain link fence to an ironclad fence all at once across the entire zona pellucida. So it becomes a barrier to preventing two sperm from fertilizing at once. But again, I digress. So as we continue with maturation, this primary follicle is going to turn into a secondary follicle, and then the theca folliculi are going to be formed, and that's going to be formed from the stromal cells. So that looks kind of like this. So here's the primary oocyte. It's surrounded again by that zona pellucida. That zona pellucida is going to be surrounded by what's called the corona radiata, which is around here. Then around that we have the granulosal cells, which are depicted in purple. What you should notice here, though, is that we're starting to get this this antrum area, this area that is starting to be filled with fluid and is not filled with cells anymore. All right, and that's going to increase as we become more and more of a mature follicle. All right, surrounding all of that, again, we have the basement membrane just like before, and then we also are going to have those stromal cells with the collagen fibers, lots of blood vessels here because we've got to get a lot of blood supply. We're going to have a lot of differentiation that's occurring very quickly, and then we here have the theca interna and the theca externa, which are what we call the theca folliculi, and that's indicating that a follicle is getting ready to enter into maturation. And this is what that looks like underneath the microscope. This is a secondary follicle. Again, here's the primary oocyte itself, surrounded by the zona pellucida, which is surrounded by the corona radiata, right? The corona radiata are going to be part of what is going to become the cumulus mass. Surrounding that, we're going to have granulosal cells that are going to be surrounding that secondary follicle. They're called secondary follicular granulosa cells. But we also have granulosa cells all around here as well. Inside here, again, this is what I was pointing out. This is the antrum. This is a cavity that's filled with fluid and no longer filled with cells. And then on the outside, we have the theca folliculi. Again, that's going to be comprised of the theca interna and the theca externa. So that theca folliculi has two different sets of theca. So in the secondary follicle, the theca is going to differentiate again into two different sections, the theca interna and the theca externa. And that very innermost layer of granulosal cells that we saw surrounding the oocyte is going to become the corona radiata. And this is going to become, or be called now, it's a mature follicle. This is a Graafian follicle. All right, so this follicle is ready to burst open and have the oocyte secreted. All right, again, so here we have the primary oocyte. Surrounding that, we have the zona pellucida. Surrounding that, we have the corona radiata. Surrounding that, we have the granulosal cells. Right? Surrounding that, we have the basement membrane. Then we have, and let's not forget here this antrum. Again, this is important because this is follicular fluid. This is not cells. And this is going to allow this to actually burst open so this can rupture off so that we can actually secrete it from the ovary into the uterine tubules, uterine tubes. Again, on the outside here we have the theca folliculi. The theca folliculi is comprised of the externa and the interna. All right, and so this is what this looks like. We are going to show you here in this ovarian cortex the oocytes that are in multiple different stages of differentiation, right? So here's this germinal epithelium. That's going to be the outside part of the ovary. And then here we have the tunica albuginea. Again, we saw that in the testes too, didn't we? Right? Then we have a primordial follicle. These guys have not started to differentiate yet. This is something that you would find even in an infant female. In a sexually mature female, however, you would also find cells that had differentiated first in these primary follicles. And then the secondary follicles, and then they would eventually be ready for a release, which would be this guy here, right? We have the secondary follicle that's going to be a mature oocyte. Here's a primary oocyte inside here, and again, we're going to have the zona pellucida around that, and then the corona radiata, yada, yada. All right, so while we're in the mature follicular state, so in the mature follicle, we, remember our oocyte is diploid, so the diploid primary oocyte is going to have to get to the haploid stage. And so we're going to have to do that through meiosis 1. right? And so in order to get to the point where we are haploid, we're going to make a haploid secondary oocyte and a haploid first polar body. So I'm going to halt this for a second and tell you that, remember when we looked at spermatogenesis and meiosis 1 and meiosis 2, we talked about the cytoplasmic bridges, how they shared everything, and how it was all hunky-dory, and that is not the case with the female reproductive system. In the female reproductive system, we make one oocyte and one polar body. And then that one doesn't turn into anything. It doesn't undergo meiosis 2. And then in the second set, we make the ovum and a secondary polar body. So only one comes out instead of 4, and we don't share anything at all, right? It's very selfish, actually. And the ovum is not going to become what we call a zygote until the fertilization event, when it actually is going to get fertilized by a sperm. So a zygote would be diploid, the ovum is haploid. So we have to, this whole slide is basically talking about how a primary oocyte, which was diploid, is going to have to undergo meiosis 1 and meiosis 2 to become the ovum. And we'll talk about that in just a second. Here we are. All right, again, I want you to pay attention to the ends and the two ends. Starting off as 2N, we are oogonium, so we are a precursor cell. As a primary oocyte, we are still 2N, so we are still diploid, and we need to get to haploid. So what happens? Well, we undergo meiosis 1, and we end up with 2 haploid cells. But guess what? Only this one gets the attention. This is the only one that becomes the oocyte. This one becomes the first polar body. Every once in a while, this first polar body also makes 2 other polar bodies. It doesn't always happen, but sometimes it does. So it may or may not divide into these secondary polar bodies. So sometimes you can end up with 2, sometimes you can end up 3. But you only ever end up with 1 secondary oocyte, right? And so then fertilization occurs. Fertilization is basically when we are going to have the sperm entering into the ovum, and then we're going to go from 2 haploid cells into 1 diploid cell here, which is the zygote. All right, and that's going to be when we are uniting the ovum with the sperm cell during the process of fertilization itself. Okay, so what's interesting about the female reproductive system is that it's not only monthly, it's actually spanned over a woman's entire lifespan. So that very first stage, where we went from the oogonium to the primary oocyte, to the primary oocyte halted in prophase 1, so we got through the very first stage of meiosis and then stopped at prophase 1. And we stopped there from the fetal development period all the way through childhood until we hit puberty, and then again it ends at menopause. And so it's actually something that spans not just on a monthly cycle, but actually on a life cycle as well. All right, so once we hit puberty, and from that point on, 1 primary oocyte, which is arrested in prophase 1 and has been for 15 to 40 years, is going to enter into differentiation to become the primary oocyte to enter into meiosis 1, okay? Sorry, this one goes to meiosis 1. This guy here becomes a polar body. And sometimes it divides into 2 polar bodies, but sometimes it doesn't bother to divide at all because it's not going to be used for anything. So sometimes it gets the clues and follows them, and sometimes it just kind of exits stage left. And this secondary oocyte here, which has now gone from diploid to haploid, now is going to undergo meiosis 2, and then enter into the fertilization event, which is going to be, the fertilization event is going to be completed as soon as the sperm cell enters into the egg cell for fertilization. All right, and that means that we're going to have that last polar body kicked off just after fertilization, and the fertilized ovum, which is now going to end up becoming, in just a second, a zygote, is going to be ready for implantation. All of this is going to occur in the uterine tubes. All of this is going to occur in the ovary. So there's a little bit of a jump that has to happen during that section, and that is actually the process of ovulation. So the process of ovulation means that primary follicle, during the process of primary oocyte here, goes to a secondary oocyte. By the time we get to metaphase 2 of meiosis 2, we're going to go to this mature follicle here. Ovulation is going to occur. We are halted at metaphase 2 until the completion of meiosis 2 occurs during the fertilization or just post-fertilization when the sperm enters into the egg. And again, this is going to happen in the uterine tubes, and this here happens in the ovary headed into the fallopian or uterine tubes. All right, so we have 2 of these fallopian or uterine tubes. They're also known as oviducts. that extend out from the uterus. And they are going to be the pathway for the egg to travel from the ovary into the uterus itself. They're also the pathway for the sperm to travel upwards to reach the ovum, and then for that fertilized ova to travel into the uterus itself. All right, so the end of the uterine tube is called the infundibulum. Funny, you've heard that before. You heard that when you were talking about the hypothalamus, right? So it's really just a structural description. And it has these projections that come out of it that are called the fimbriae. And the widest portion of that tube is called the ampulla. So the ampulla is the region that's going to be here. It's going to basically going to be the region where hopefully we already have fertilization occurring and we have a fertilized egg that is rolling down here that is ready for implantation. All right, so what do we see here? This is the ovary, right? The ovary is going to release a mature oocyte here into the infundibulum of the uterine tube and then head into the ampulla region of the uterine tube. From there down into the isthmus, which is going to enter into the uterus. Now, you've seen the word fundus before. Again, fundus just means opposite to the opening. So if you consider that the vagina, or really the cervix, is the opening, then the fundus would be the opposite side. And in pregnancy, what they're talking about is the fundal extension. They're talking about how high the fundus of the uterus goes up into the abdomen, which indicates, or can indicate, how far along you are during your different stages of pregnancy. Inside this uterus right now, we have nothing. It's just a uterine cavity, right? We're going to hopefully be bringing in a fertilized egg to implant here, and we have several different layers. So we have the endometrium, which is the top layer, the myometrium, which is the middle layer, and the parametrium, which is this bottom layer. And ladies, during your female cycle, when you are sloughing off the cells, they're coming from this endometrial layer here. So this endometrial layer is going to become flush with blood once a month for 3 to 4 days, and then it's going to all slough off afterwards if we do not end up with a successful implantation event. This here is the internal os. We also have an external os. So this is the cervical canal. So the cervix is going to be the very tip of the uterus that connects to the vagina. Here we are in the vagina itself. The vagina is has these rugae inside of it, which you've seen in several other descendable organs like the stomach and the bladder, and so the rugae is going to allow the vagina to expand, and then the sperm are going to be deposited likely somewhere here towards the end of the vagina and have to get through some of the vaginal secretions and then also through the cervical mucus and then enter into the uterus, then swim up through the uterus, then go through the isthmus, then go through the ampulla, and somehow somewhere in here meet up with the egg, which has been released by the ovary into the infundibulum and is making its way that direction down into the ampulla. All right, so I feel like I've touched on a lot of this. I haven't really touched, yeah, I think I've covered that. All right, so the uterine tubes are going to have 3 different layers. These fallopian tubes have 3 layers, the mucosa, the muscularis, and the serosa layer. The mucosa layer is going to have a simple ciliated columnar epithelium. Basically, we have cilia that are in there that are going to help move the fertilized ovum, or the secondary oocyte, as it may be, towards the uterus itself. So this is basically a little set of tiny little fingers that are one at a time pulling this hopefully fertilized ovum down into the uterus itself until it reaches an area that's nice and cozy and it can implant itself. There are specialized cells called PEG cells in this tube, and they're going to secrete specialized fluids that are going to be providing nourishment for the ovum itself. And so that kind of looks like this. These are the PEG cells here. They're non-ciliated, but they do have microvilli on them. These are ciliated cells, the simple columnar ciliated epithelial cells. They look like this, like little teeny tiny finger projections. And they basically are moving the egg along until it finds a suitable location for implantation. All right, so that's going to occur in the uterus. The implantation event's going to occur in the uterus. The uterus is obviously a part of the pathway for sperm to travel through, and then they travel up into the uterine tube where a fertilization event occurs. And then the implantation of the fertilized ovum is going to occur inside the uterus itself. The uterus is also going to be the location where we're going to sustain the fetal development throughout the pregnancy, and also obviously the organ that's going to be responsible for labor and delivery. As we mentioned previously, and again, we've seen fundus several times. We talked about that in the gallbladder. We've seen that in the stomach. But it's going to be the region that's going to be opposite the opening. So the top of the uterus is called the fundus. The central portion is called the body, and then the region that connects into the vagina is called the cervix. There's also a little bit of a region that's called the isthmus that's going to be between the body and the cervix itself. Inside the uterus is the uterine cavity. interior of the cervix is called the cervical canal, and the cervical canal has two ends. The opening of the canal is the internal os, and the other side of the opening that enters into the vagina is the external os. So the part that is going to line up into the uterus is internal, part of the vagina is external. So the uterus is going to have three layers. The serosa layer is called the parametrium, that's the outermost layer. The middle layer is called the myometrium, and the myometrium has multiple layers of smooth muscle, which are going to help for contraction during delivery, etc. And the endometrium is that inner layer. The endometrium has a layer called the stratum functionalis, and that's going to undergo multiple sets of changes, but what really it's going to enter, it gets thicker, it has a lot of vasculature, etc. It basically is getting itself ready, making a bed for a nice happy little layer for that egg to implant into. And if the egg doesn't implant, then the stratum functionalis layer is going to be shed during menstruation. So the layer directly underneath that is called the stratum basalis. And the stratum basalis layer is going to be permanent, and it's going to be where the stratum functionalis comes from after each menstruation and when we're starting anew for the cycle. Okay, so if we are looking here at the second week of the menstrual cycle and the third week of the menstrual cycle, we're going to see a huge difference in the stratum functionalis. But if you look, the stratum basalis looks the same, right? This basal layer, which is what all of this comes from. So the endometrium, generally, when we are not going to be ready for implantation, is going to be kind of shallow here, right? Not going to have a whole lot of that stratum functionalis section. But when we are ready for implantation, we have all of the stratum functionalis ready, which is going to be highly vascularized, and it's a nice comfy bed for a ovum to implant into. Underneath that, again, we have the stratum basalis and the myometrium. Those layers do not change throughout the weeks of the menstrual cycle, at least not dramatically. Again, the internal region is going to be the lumen of the uterus. Here we have a columnar epithelial layer, and then the stratum functionalis here. This here is an endometrial gland, so we're going to have secretions that are going to come out of there. And then this, again, this is a stratum basalis, or the layer that is not going to change very much. Now, this whole region is going to be highly vascularized. So the vasculature is going to be coming off of the internal iliac artery. And we have these regions that are branched off called uterine arterioles that are going to supply blood directly to the uterus. And then those are going to kind of come together, these uterine arteries are going to come together to arcuate arteries that are going to feed into the myometrium. Then they branch out, they form these radial arteries that go very deep into the myometrium. And we also have another set of arteries that are straight arterioles that go directly to the stratum basalis. So as I mentioned, it's a highly vascularized system, and this is why uterine hemorrhage used to be one of the major causes of death during childbirth before we had modern medicine and intervention. So this is just an overview. If you're looking at the uterus with the partial section taken out here, the top layer here, this is the perimetrium, that's that outermost layer. Then we have the myometrium, which is the middle layer, and that central layer is the endometrium. This is what's going to get larger and smaller throughout the month, right, when we are going to have that nice bed made or versus having sloughed it all off. Inside here we have the uterine cavity, and then all of this is fed, as I mentioned, here's the uterine arteries on either side, which feed into these arcuate arteries. We also have radial arteries. All of these are going to feed into the different regions, right? So the radial arteries are going to go all the way down into the myometrium here, right? So, okay, so the cervix is going to also be secreting things. It's going to produce cervical mucus. You may have heard me saying that's one of the first barriers that the sperm have to overcome to be able to fertilize the egg. So the cervical mucus actually changes throughout the month. When it is more hospitable to sperm is when the best chances of fertilization to go right are, right? So it's when we have the highest chances of a fertilization event going correctly. And so we have chemical changes that occur during the time frame when the sperm are most likely to be able to fertilize the egg. So it's going to be a little bit less viscous, a little bit more alkaline, so it's not going to be very acidic. It's going to help nourish the sperm. And there's also a process called capacitation. So just like the sperm had to undergo a maturation event inside the epididymis, They also have to undergo a maturation event that occurs inside the female reproductive tract. So you can picture this like people, if you were considering these guys as soldiers that had to all get to the end goal, right? And the uterine tract was actually villagers that were handing the soldiers Gatorade on the way, right? Helping to aid them in their ability to fertilize the egg. And this only happens, or it happens best, during particular times of the month. So these functional changes in the sperm are going to allow them a better chance to fertilize that secondary oocyte. So the vagina itself is a canal. It's muscular. It's going to be lined with mucus, and it's going to extend from the exterior to the cervix. You also notice it has a lot of rugae in there, so it's able to extend and distend. It is connected here up to the uterus by the cervix. It's also going to lie just posterior here to the urinary bladder. So the vaginal mucosa is going to line continuously in to that of the uterus. So the lumen of the vagina, if you're going to take a look at it, it's going to have the mucosal cells. These mucosal cells are stratified squamous epithelial cells on this layer here. And then underneath that we have the lamina propria, right? And then if you continue downward, we have the outer circular layer and the inner longitudinal layer. And so these are going to be the layers of the mucosa of the vagina. So this is the lumen, so this would be the central part of the vagina itself. Okay, so we have a lot of connective tissue in the vagina, and these are going to be found in these folds that are called rugae. Again, this allows the vagina to stretch during intercourse, and then of course also during childbirth. So these rugae allow for drastic changes in shape without distortion or ruining the organ itself. We have a muscularis layer that's comprised of two different regions, an outer circular layer and an inner longitudinal layer. The inner longitudinal layer is comprised of smooth muscle, and again, all of this is going to allow the vagina to stretch quite drastically. Now we have a very thin fold of mucous membrane that's highly vascularized. It's called the hymen, that's going to close off the inferior end of the vagina until puberty or intercourse or some sort of traumatic event. So this is an overview of the external region of the female reproductive system. This here is the mons pubis. This is the prepubis of the clitoris, also known as the clitoral hood. Underneath that is the clitoris itself. These here are the labia majora. They're actually spread out here. These guys here, these inner folds, are the labia minora. As I mentioned, the female reproductive tract is going to have separate tubes than the female urinary tract, which is not the same for the male. So this here is the external urethral orifice, which is found between the clitoris and the vaginal orifice. And then this little region here is the hymen, which is a little region of tissue which is highly vascularized and can be torn at the very first sexual event and tends to release blood at that point. All right, so we're looking at the external genitalia of the female, where you can consider the vulva, which is going to include the mons pubis, the labia minora, the labia majora, the clitoris, and the vestibule. The vestibule is going to be basically the entrance, so that's going to include the hymen, the vaginal orifice, the external urethral orifice, and a bunch of different glands are going to have ducts that open there as well. Those ducts are going to include Skene's glands, called the paraurethral glands. These are going to secrete mucus, and they're found embedded into the wall of the urethra itself. So they are homologous to the prostate, and they secrete into the urethra. So they don't secrete into the vagina, but they do add to the secretions during excitement and intercourse. So they also have the vestibular glands. These are called Bartholin's glands. These are going to be mucus-secreting glands. They are going to end up adding lubrication during sexual arousal and are homologous to the bulbourethral glands, which are found in the males, which means that they arise from the same subsets of tissues during differentiation and development. Okay, so the vestibule bulb has two masses of erectile tissue that are going to actually engorge during sexual arousal, so they're very vascularized, and they actually tighten down the vaginal orifice, and it's going to be similar or homologous to the erectile tissues of the penis. So they're highly vascularized, they're highly sensitive, they are going to respond to stimulation with increased vascularization. All right, so if we look at the top, here is our pubic symphysis. Here's the clitoris. If If you follow your way down here, we have the external urethral orifice, the vaginal orifice, and then behind that here, we're going to have several sets of muscles. So here's the ischio-cavernous muscle. Here's the bulbospongioso muscle, and the peritoneal muscle. And these guys together form the urogenital triangle. Okay, and the urogenital triangle sits directly on top of the anal triangle, so it uses those same muscles, the peritoneal muscles, to form in this case, the top of their triangle, but the bottom of the urogenital triangle, and connect down here at the coccyx to the gluteus maximus, and all of that is going to surround here. This is the anus. Okay, so let's just summarize some of the male and female reproductive structures that are going to be similar or homologous, meaning they came from the same subset of cells during differentiation and development, just given different context clues. So, for example, in the female, the ovaries are very similar to the testes, the ovum or the egg to the sperm itself. The labia majora is going to be comparable to the scrotum, which is what would be holding the testes, except the testes are actually ovaries, so they're up inside instead of descended. The labia minora is going to be the spongy urethra. The vestibule is similar to the intermediate urethra. Also, the bulb of the vestibule is going to be very vascularized, so it's going to be similar to the corpus spongiosum of penis and also the bulb of the penis. The clitoris is similar to the head of the penis or the glans penis, and it also is going to have a corpora cavernosa inside of there. The periurethral glands are similar to the prostate, and the vestibular glands are homologous to the bulbourethral glands. So do take a good look at these structures. There are a couple questions on the exam that are going to ask you specifically which of them are homologous, or I think more specifically which of them are not homologous. Okay, so let's talk about lactation for a minute, right? So we have specialized glands in the female that are found underneath the breasts that are actually modified sweat glands. So they're modified sudoriferous glands that are actually modified to produce milk, and they have a lot of different lobes, so 15 to 30 different lobes that are comprised in a ring that has lobules that are going to contain milk and secrete that milk out through these glands that are called alveoli. And at the end of the breast we have a nipple that's going to be where the milk is going to come out, and there's going to be specialized ducts at that nipple. They're called lactiferous ducts, where the lactation is actually going to occur. There's also going to be a dark skin that's going to be kind of dense and going to be able to withstand like an infant's teeth, for example, so it's a little bit less sensitive than some of the other skin, and that's going to be called the areola. So this is a sagittal section of a female mammary or breast tissue. So here we look, they have the ribs. Inside the ribs we have the intercostal muscles, right? And then we are going to have here's the pectoral muscles, so those are actually found underneath the breast tissue. Then we have the pectoral fascia, which is going to be the connective tissue that lines on top of the pectoral muscles. Okay, so now we can get into the breast itself. As we move forward, we have all of this yellow is going to be adipose tissue or fat tissue that's going to be in a sub-Q layer, and that's going to surround these specialized lobules that have alveoli in them. These alveoli are going to feed into secondary tubules, into mammary ducts, and then out through the areola, through the nipple, which is surrounded by the areola, out through the lactiferous ducts and the lactiferous sinus of the nipple, again surrounded by the areola. And these are all the same, just from a different perspective. Okay, so females that are not pregnant are going to experience cyclical changes in both the ovaries and the uterus. And it takes about 1 month. Some take a little bit longer, some take a little bit less. But if you end up pregnant, then the cycle is going to be interrupted and sustained by various hormones that would normally be cyclical that are instead secreted constantly. All right, so the cycle is going to involve an entire rotation from oogenesis, and also the preparation by the uterus. So we have two different organs that need to work in concert, right? The ovaries are going to have to create and mature that egg, and then also the uterus is going to have to end up with that really nice, lush bed in the endometrium. And this whole thing is an ovarian cycle, and it's going to be in concert with the uterine cycle. So the ovarian cycle, again, that's going to include everything that's involved in the maturation of the oocyte, and the uterine cycle is going to involve anything that has to do with the changes required for the endometrium to be able to accept the implantation of the developing embryo. So something called GnRH, or gonadotrophin releasing hormone, we may have heard of before. In fact, we heard about it when we talked about male differentiation and development. But that's going to come through the hypothalamus, and it's going to be responsible for the control of both of these ovarian and uterine cycles. And this is important because obviously you want these guys to be it's cycling at the same time, because if their windows aren't lined up, then we can have issues with fertilization, right? So this looks similar to what we saw in the male, in that the fact that the GnRH is going to be responsible for stimulating FSH and LH, which previously was going to be combining with testosterone in the male, but here in the female we don't have as much testosterone, and it's not going to work in the same pathway. So that's about where our similarities end here, is that the GnRH is going to be released from the anterior pituitary and stimulate the release of FSH and LH. But FSH and LH now have different roles. So FSH in the female is follicle stimulating hormone. It's going to stimulate the development and creation of the ovarian follicles. It's going to make them grow up to be able to be secreted by the ovaries. Okay. It's also going to have development of the ovarian follicles and secretion of particular things, including estrogens and inhibins. All right, so hold that thought, because we're going to get back in a minute to what estrogens and inhibins do, but I'm going to continue on with my thought of the fact that FSH and LH are going to be secreted from the anterior pituitary, and that LH is also going to help stimulate things in the ovaries, including ovulation event. All right, so FSH is going to stimulate the growth of the follicles. LH is going to stimulate the actual rupture and ovulation. And that also is going to stimulate the corpus luteum, right? Which is going to then create secretion of certain things: progesterone, estrogens, relaxin, and inhibins. So after we have had the egg released, we are still going to have a role that's played by what's left of that follicle, that corpus luteum, which is going to secrete things like progesterone, estrogen, relaxin, and inhibin. All right, so these are the four main hormones that we're going to talk about. So I'm going to walk you through them, and then we'll walk you through how they work in concert. Let's start with estrogens. All right, estrogens are responsible for the maintenance and development of the female reproductive structures. Also, all of your secondary sex characteristics like breast development and puberty, etc. It also is going to help increase protein anabolism. So anabolism is putting things together. Catabolism is breaking things apart. So we are building up proteins. Also going to help with lowering blood cholesterol. Is going to inhibit the release of GnRH, FSH, and LH. Where did we see that before? Oh, in the male reproductive cycle hormonal regulation. So it's going to hit up in the hypothalamus and in the anterior pituitary, where it is going to be its inhibitor of GnRH. Okay, so all of these are going to be these kind of feedback loops. But remember, these guys are released cyclically, so there are going to be moments when we have less estrogens, and so we're going to upregulate the GnRH. But I digress, and we're moving on. So here's progesterone. Progesterone works in concert with the estrogens, and it's going to help prepare that endometrium for implantation. So this is going to help make the endometrium nice and thick, a nice uterine lining for that egg to bury into. It also is going to prepare the mammary glands for lactation and inhibit the release of GnRH and LH. So again, now we have an inhibitor of what it was initially being stimulated by. So it's a feedback loop there as well. So here's relaxin. Relaxin is going to be responsible for inhibiting contractions of the uterine smooth muscle. And during labor and later on in late pregnancy, it's also going to increase flexibility of different regions, including the pubic symphysis and dilates uterine cervix. As a side note, this can also cause women's ankles and joints to get very, very flexible, so they might be more likely to twist an ankle or twist a knee, or for their shoulder to come out of socket or something like that, because this relaxin is going to affect all of the different bone flexibility, basically, joint flexibility. So moving on to inhibin. Inhibin is going to inhibit the release of follicle-stimulating hormone and also luteinizing hormone. So this is also another way that we are regulating, in this place at the anterior pituitary gland level, regulating the hormonal release. Okay, so I know that was a lot. Don't worry, we're going to talk about it from a couple other angles as well. So the female reproductive cycle is generally going to range from 34 to 46 days and is comprised of 4 major phases. The first is the menstrual phase, which is the one whereby everybody kind of knows. So this is where we're going to set our clock, is by the first day of menstruation. After the menstrual period, we have the pre-ovulatory period, then we have the ovulation cycle, and then the post-ovulatory period. All right, so we have 2 things that we're going to be paying attention to in terms of the female reproductive cycle. This is the ovarian cycle here at the top and the uterine menstrual cycle here at the bottom. And these are kind of shown overlaying because they are both happening at the same time, hopefully in concert. So in the ovarian cycle, the first thing that has to happen is that follicular phase where the primordial follicles are going to then become the primary follicles, are then going to become the secondary follicles, and eventually mature follicles, and eventually ovulate, right? After the ovulation event, this empty follicle is called the corpus luteum, which eventually becomes the corpus albicans, and that's going to secrete a bunch of hormones as well, including progesterone and estrogens. All right, so the estrogens that are secreted here by the secondary follicle and the mature follicle during and up through the ovulation event are going to increase the proliferation of that stratum functionalis there in the endometrium. Okay, so the stratum basalis, again, that's going to maintain the same for most of it, but the stratum functionalis is what's going to rise up, become very blood-filled, become very nice thick bed, and then slough off during menstruation. So if we start off with menstruation as day 1, right? So we slough off all that fluid, and then starting around day 4 or so, we're going to start proliferating again. So we're getting a larger and larger stratum functionalis, and that's happening at the same time that the ovaries are allowing the follicle to mature. Again, just one is happening from either or the other ovary. Then that mature follicle is going to ovulate. Ovulation should happen right around the same point that we have the thickest endometrial layer, right? And this layer is maintained by the secretion of progesterone and estrogens. Now, if there is no implantation occurring, then this cycle is going to end up ending at menstruation, and these are not maintained. If, however, we do end up with an implantation event, progesterone and estrogen are going to maintain this nice thick uterine lining for that newly developed or developing embryo. Okay, so let's look at it from another angle. And this one I actually do want you guys to know. This one is going to be on the exam. So just be aware of maybe not all of the details, but of which part of the cycle each of these hormones come into play, right? So this is obviously hormone concentration, so the higher this is, the more of the hormone at that time. This here is the days of the female reproductive cycle. Kind of early on, we're going to start stimulating that follicle with FSH, and then we get another surge of FSH right around the same time that LH, or luteinizing hormone, is also secreted. Okay, this is going to be right around ovulation. These estrogens are going to increase right around the point of ovulation and then drop right off, and then come up a little bit again towards the end, right before we have our actual menstruation again. Progesterone, on the other hand, progesterone, remember, is secreted by the corpus luteum, which is that empty egg follicle after the egg has already been secreted, so it doesn't start until a little bit later, and this is going to either stay on if pregnancy occurs, or then get lowered and turned off if pregnancy does not occur, which leads to menstruation. So that's kind of an overview there of the different hormones involved in the female reproductive cycle. And as you may have noticed, all of these hormonal cycles are really tightly regulated by negative feedback loops. And so you want to make sure that you are never having too much or too little. You want to make sure it's all in concert. And so there's a lot of regulation that has to be tightly fine-tuned. And that means that the different players, things like estrogens, are going to have positive and negative feedback loops on different parts of the cycle. So for example, high levels of estrogens that are found in the last part of the pre-ovulatory phase are going to positively regulate the cells that are secreting LH and GnRH, and that's going to cause ovulation, because you want to make sure that you are keeping your uterus and your ovaries kind of communicating, right? And so there's a lot of different hormonal interactions that are found between the ovarian and the uterine cycles, so that we can make sure that our timing is lined up, because remember, this is a very precise window that we have. It's only in the fallopian tube for a very short period of time. Okay, so here, this is what I was talking about with the high levels of estrogen. So they're going to come from the mature follicles, or the almost mature follicles, and that's going to stimulate the release of more GnRH and LH. So the mature follicle or premature follicle is going to have a positive feedback loop where it's going to secrete more of the GnRH and the LH, which is going to increase its ability to eventually become ovulated, okay? Because that LH surge is what's going to cause it to rupture. So this is one of the very limited examples of a positive feedback loop that we'll discuss in our anatomy classes. All right, so we're going to toss in all of the information that we've shown you in the past couple of slides. So I know this shows a lot, but if I were you, I would use this to study from. I would pop this figure up and start talking your way through it and say, I understand or I don't understand. And obviously, if you don't understand, I would work backwards to the slides that were a little bit more detailed. But this is kind of an overview. So again, the information coming from the hypothalamus, the hormones coming from the hypothalamus go through the infundibulum into the anterior pituitary. That's going to include things like FSH and LH, right? Follicle stimulating hormone, luteinizing hormone. Follicle stimulating hormone is involved in the stimulation of the follicles. So the follicles are going to grow from primary and secondary follicles and eventually become matured. LH, on the other hand, is responsible for ovulation. So that surge in LH is going to increase the chances of ovulation until eventually it ruptures. And then remember, the corpus luteum, which is what's left, is going to be secreting hormones that are going to help maintain this endometrium. And eventually it becomes the corpus albicans. Okay. At the same time, so that's all what's happening in the ovary. At the same time, in the uterus, we're going to have a little bit of menstruation for the first couple of days, and then repair and proliferation of the uterine endometrium. So proliferation of the endometrium is going to make it nice and thick and ready to accept an implanted zygote. So we are preparing the endometrium for the arrival of the fertilized ovum, and that is going to be sustained here by secretions of the corpus luteum. And if that is not the case, then it's not going to continue to secrete the progesterone. It's going to stop secreting progesterone, and menstruation is going to occur, and we are going to lather, rinse, repeat. And again, we have several different negative and positive feedback loops here. So one of the negative feedback loops is here we're going to be regulating the GnRH and the FSH that is released from the hypothalamus and the anterior pituitary here from some of the ovarian hormones. But at the same time, if we have high levels of estrogen in the absence of progesterone, it's going to increase the release of these same hormones. So it's a very complex system whereby we have both positive and negative feedback loops that are keeping the hormonal regulation both tightly maintained and also cyclical in nature. All right, so let's talk about birth control for a moment here. Right? So birth control methods basically is just defined as anything that is attempting to limit the number of children produced by preventing conception. So that's going to include everything from abstinence to abortion. So complete abstinence means that we are never going to end up with sperm and egg ever meeting. And then we have a ton of other methods. So some methods include surgical sterilization, for example, having your tubes tied or having a vasectomy. We also have hormonal methods. So if we stepped backwards and looked at this female reproductive cycle, you can see that all of these different hormones here are turned on at specific times. So if you can trick your body into maintaining this without being pregnant, it'll already think that it's pregnant. So if you keep progesterone on for a longer period of time, it'll never ovulate again because you think that you're pregnant already, for example, which is a very simplified version of how the how the birth control pill works. But some of the other methods include periodic abstinence, so only engaging in sexual intercourse during days that are less likely to result in pregnancy. This is also known as the rhythm method. But everything other than complete abstinence is going to have a slight failure rate. Some of them are going to have much larger failure rates. For example, the rhythm method is going to have one of the largest failure rates. Let me verify that's true. Well, anyway, so if you take a look, complete abstinence means that we have zero conception. Surgical sterilization for the males is a vasectomy, where we are going to ligate the vas deferens. And you would say, well, that should be 100%, and it should be. However, there is a 0.1% chance that the tube will heal itself, regrow, and allow sperm to come through. And so that is very low risk, but is still a possibility. And the same thing with the tubal ligation. So having your tubes tied means that you're going to have those fallopian tubes. Generally, they'll sever them, or they'll just wrap something around it, or sometimes they'll have something called scarification, where they will go in and damage it such that nothing can get through it. But sometimes the body heals itself, and so you have a 0.5% chance that a tubal ligation is going to heal itself and allow the egg to meet the sperm. Some of the hormonal methods are going to include things like birth control pills, lots of different types of birth control pills. We also have non-oral contraceptives, so now we have a patch that women can take, a contraceptive ring that goes inside the vagina, and then we also have other types of emergency contraception, like well, for example, condoms, for example. We also have Plan B pills. Also, there are hormone injections that you can give yourself, particularly women at different stages, that will prevent you from ovulation as well. But as you can see, all of them, although they have low percentage pregnancy rates, there are still a very slim percentage chance that you could end up pregnant during these. So here's some of the other birth controls that people use. Intrauterine devices are copper, the Mirena is one. There are several different types. And again, with perfect use, you have approximately 0.6% chance of having a fertilization event or having a pregnancy occur. Spermicides alone, you have 15% chance with perfect use and almost 30% chance throughout 1 year, right, for unintended pregnancy throughout 1 year of use. So you have almost 30% chance of an unintended pregnancy throughout that year. So chemical methods alone are really not super efficient. Some barrier methods include condoms, right? Male condoms, vaginal pouches, a diaphragm, which is going to pop over and cover your cervix. So you can have that with or without spermicide, but generally with spermicide is how they do it. We also have a cervical cap, which is a little bit different from a diaphragm. You get fitted for it specifically. And all of these have a failure rate of between 5 and 10% with perfect use and up to 20% with typical use. So again, there's always going to be a chance that fertilization is going to take place. And here's what I was talking about with the rhythm method or the periodic abstinence. So some people will do it by the rhythm method, and then this is a symptothermal method whereby they actually take the woman's temperature every day, and during the days of ovulation, you increase, or women will increase in temperature by just a very small amount, but if you take it every day, you know which days to avoid. And so that does decrease the percentage risk by a little bit, from 9 down to 2%, but again, you do still have failure rates, and again, this is perfect use. If you look over a typical use, it pops up to 20 to 25%. And with no method at all, with 2 viable, healthy, fertilization age adults, we have approximately 85% chance over 1 year of an unintended pregnancy with regular sexual intercourse. All right, so we talked about different methods. Again, the surgical sterilization would include things like a tubal ligation or a vasectomy. Vasectomy is for males. We remove a portion of the vas deferens. Sometimes they just clip it. Sometimes they pinch it off. Sometimes they burn it. But either way, you're going to remove a section of it. In both of, obviously, you'd have to do both sets of them. And then the tubal ligation is used in females, and you tie off and then snip the uterine fallopian tubes. And then another method for females is a non-incisional sterilization, and it's called scarification, and they send in a long coil that has sharp metal and plastic into each of your uterine tubes and kind of twist it a little bit until it ends up damaged, and then scar tissue grows in and blocks these tubes. These are very delicate structures with very small openings, so it's pretty easy for that scar tissue to grow in and block the tubes. We also have hormonal methods. Hormonal methods are usually going to be a good option if you intend to get pregnant later. So some of the other options are going to be more terminal. Things like a vasectomy or the scarification are going to be very difficult to reverse, perhaps. But the hormonal methods are things that you can go on and off as you decide to do your family planning. So lots of different types of oral hormonal methods, or the birth control pill. Generally, they're going to contain different levels of progestin and estrogens that you take throughout the month. Some birth control pills are going to contain both of them, and you will very rarely actually menstruate. You'll menstruate only approximately every 13 weeks or whenever the pills set you up to menstruate. And then there's other types of pills that are lower estrogen, like mini pills that contain progestin only. And progestin does a couple things involved in regulating the cycle, but it also is going to involve in thickening the cervical mucus, which is one of the barriers that sperm have to get through to penetrate to be able to get to the egg. Some other contraceptive methods that are non-oral include a contraceptive skin patch, a contraceptive ring that goes inside the vagina. So these are all hormonal methods that are non-birth control pills. Also emergency contraception, which is actually an oral method because you do take the pills orally, but you don't take them for a longer period of time. You take them 72 hours apart. 72 hours in the 72-hour window after unprotected intercourse, and then 12 hours from the next. And usually they're just high levels of estrogen or progesterone meant to disrupt that cycle if fertilization did occur, so that implantation does not occur. Barrier methods are meant to make it that sperm and egg don't actually ever interact, so it's a physical barrier that keeps sperm from gaining access to the uterus and therefore the uterine tubes. And that's going to include things like the condom. There's a female condom as well. It's called the vaginal pouch, something called the diaphragm, and a cervical cap, which are both going to be in between the vagina and the cervix. And one is going to be fitted specifically for your cervix, so the cervix needs to be fitted for specifically. Also, as we talked about, abstinence is when you are just simply not having a sexual intercourse for a specific period of time. This used to be known as the rhythm method, and this is when you are abstaining from intercourse in time frames when ovulation is most likely to occur. And this is really employed for people who have a good knowledge of the different physiological changes that take place during the female cycle. So you actually have to have discipline as well. And some things that will help people who are employing the rhythm method are going to include taking the temperature of the woman in the morning, as well as, so symptom meaning looking at symptoms, thermal meaning looking at temperature, as well as looking at the cervical mucus, so looking at the thickness of the mucus, the color of the mucus, the scent of the mucus, etc. So we're looking for specific markers that will say this woman is ready for ovulation, so now would be a bad time. So abortion is when we are going to eliminate a product of conception from the uterus. So this is a post-conception event. It can be pre-fertilization or post-fertilization. I'm sorry, it can be pre-implantation or post-implantation. It is always post-fertilization. So mifepristone is a hormone that's used in the early stages of pregnancies, about 9 weeks or less. And it's going to just cause menstruation. And then a physical abortion usually uses a vacuum, so a suction aspiration to be able to remove the embryo or the fetus, and generally also the placenta and the uterine lining. And please keep in mind when we talk about the word abortion, I know it's in the news a lot recently, but abortion refers to the removal of fetus and fetal byproducts. So it also is going to involve anyone who is already had a miscarriage event but has not expelled that tissue yet. That is still considered to be an abortion even though it is not alive, and so this is something that is commonly used as a medical treatment for women in that particular situation. I am going to move on here. We'll talk about a dilation and evacuation. So a dilation and evacuation is basically it used to be called dilation and curettage. It's a D&E, and it's going to be a way that we dilate the cervix up and out, and then we use suction and forceps to remove all of the uterine lining. And this can be a procedure that is performed on a pregnant woman or non-pregnant woman. So sometimes you might need to have this occur on someone who is not pregnant because they have endometrial tissue that is still wrapped around something like a uterine fibroid, for example, or they still have placental tissue post-pregnancy that was never released. So it is a procedure that is performed on someone who has something that needs to be removed, which may or may not include fetus or fetal-associated tissue. Something called a late-stage abortion is going to use a saline solution that is going to have a induction of an early birth event. Sometimes they also use surgical methods to endorse it. Late-stage abortions are typically used in cases where there are severe fetal abnormalities, or the death of the fetus is imminent, or sometimes the fetus has already passed and they still need to remove. Again, fetal-associated tissue is still considered an abortion, so this is oftentimes a harrowing surgery on women who actually wanted these pregnancies. Okay, so I'm going to talk very shortly about the development of the reproductive systems, but we're going to gloss over it and just kind of lightly touch on it because I know we've been here for quite a while now and development is its own lecture. But the gonads, which are going to be the ovaries or the testes, are going to come from a mesoderm, the intermediate mesoderm, that's going to give rise to what's called the gonadal ridges. And in the male reproductive system, we make Wolffian ducts or mesonephric ducts. And in the female reproductive system, we make Müllerian ducts or paramesonephric ducts. from the same sets of tissues. So this is an overview. Again, I'm not going to quiz you on any of this, but just so you can see here that these ducts here, the mesonephric ducts, are what's going to, oh, this is the urogenital sinus, it's all going to end up developing into either in the paramesonephric ducts here in the males or the mesonephric ducts here in the females. And so differentiation of the male and the female development occurs pretty early on and from the same subset of tissues coming from this gonadal ridge here. Okay, so if we have male development cascade initiating, we're going to have something called MIS, which is going to inhibit the Müllerian ducts. So it's going to cause apoptosis of the cells in the Müllerian ducts, so we're not going to be able to make anything from those ducts in the male. And in the female, we'll be making the Müllerian ducts because we don't have the MIS. In developing males, we're also going to be secreting testosterone in higher levels from the interstitial cells in the testes, and that's going to cause the differentiation of the male development cascade. That's going to come from what's called the mesonephric ducts, and that's going to turn into all of these male organs that you know and love, the epididymis, the vas deferens, the ejaculatory duct, and the seminal vesicles. So prior to our differentiation event, all embryos are going to have the same sets of tissues. We have the urethral folds or the urogenital folds, the urethral groove, the genital tubercle, and the labial scrotal swelling, which will turn into either the labius or the scrotum, labia or the scrotum. So this is the undifferentiated stage, about 5 weeks. Here we have the genital tubercle, the urethral folds, the urethral groove in the center of that, this labial scrotal swelling, the peritoneum, and then the anus. As we enter into male or female development cascades, this genital tubercle is going to become the glans penis or the clitoris, so it's either going to become the top part or kind of shrink down underneath the clitoral hood, which is going to be very similar, of course, to the foreskin here in the male. This urethral folds here are going to be similar in the male and the female for a minute, and then eventually these are going to seal up in the male and remain open as a external orifice, vaginal orifice for the female. So you can see that this becomes just this line here that connects in the male. It is not open, and it leads obviously directly to the anus here. That's the peritoneum, is the connection here between the anus and in the woman the vaginal opening, and in the male the urethral folds just kind of close together. This labial scrotal swelling becomes, as you might guess, either the labia majora and labia minora or the scrotum, which is going to hold the testes and the epididymis. Okay, so the juvenile stage of reproduction occurs all the way up until we hit puberty. And puberty actually starts a little bit earlier than you might think. So puberty actually starts around age 10, although most of your characteristic traits of puberty don't really occur until 12 to 15. In puberty, we're going to have a lot of hormonal changes that are going to change our outward appearance or our phenotype. So males are going to also start producing sperm. Females are going to enter into something called menarche, which is the beginning of menstruation. So they're going to have their first menstrual cycle. Interestingly enough, that first menstrual cycle is probably not the first time that that female has ovulated. So there are cases of women who end up pregnant prior to entering into menarche because they have had sexual relations prior to having the opportunity to actually have their first menses. So ovulation actually occurs prior, or a couple of months prior to menses itself. As ladies get older, and men as well, your fertility is going to decline, but particularly with women around 30 to 40, your ovarian follicles are going to start becoming exhausted. Remember, they've been primed now for 30 years. Our estrogen levels are also going to decline. And so during this time frame, this is an age where women are going to become vulnerable to what's called the maternal aging effect, where they may end up with reproductive disorders in their offspring. And so they're going to be tested for a lot more things than someone in their 20s might be tested for in terms of fetal abnormality, particularly for things like trisomies, like Down syndrome. In males, however, reproduction is possible much later, around 80 or 90 is the latest that we've had males that are producing offspring. However, around 55, the testosterone levels do decline and the sperm levels do drop. We also end up with things like fractured sperm, low sperm motility, etc. Additionally, sexual desire can sometimes wane off. Erectile dysfunction can sometimes occur. And as you get older, passing the 60 mark, you have a higher than 50% chance of prostatic hypertrophy. Basically, the prostate is going to get larger. It's going to get 2 to 4 times larger than normal, which is going to prevent things like urination and can cause problems with ejaculation. Okay, so that is going to bring us to the end of our reproductive cycle. So I appreciate you guys bearing with me for such a long-winded lecture. Have a great night.